Hello dear student, today I am going to explain you about histamine. In previous video I have started a new topic called AUTOCOIDS, and in last class I have discussed about what are AUTOCOIDS, why they are called AUTOCOID, what their mean is, what is the definition of AUTOCOID, what are their classification of AUTOCOIDs, what are their mechanism of action, what are the example of AUTOCOIDs, their specific site, and some general action of all those AUTOCOID. From today, as I promised in last class, that from today we will discuss individual AUTOCOIDs which are very important in our body, which plays an important role in doing or in pursuing or in performing many functions in our body. That all AUTOCOID individually I am going to explain from today's classes. So today is the topic that I am going to discuss is a AUTOCOID which is very important in our body, that is histamine. So histamine is a topic for today. Histamine is a AUTOCOID. I have told you in previous class only, when I have discussed about the classification of AUTOCOID, I have told you about amino derived AUTOCOID. Histamine is one example of that amino derived AUTOCOID. So histamine is a AUTOCOID which is amino derived, which got derived from an amino acid. That amino acid is histidine. So as this AUTOCOID is came or derived or belongs from a amino acid called histidine, so it is also a amino derived AUTOCOID that is named as histamine. That histamine plays an important role in our body in performing or pursuing many physiological actions that I am going to discuss into the next slides. So it is a simple structure of histamine that you are going to study in medicinal chemistry. I just will tell you about that short structure so you can better understand about its property later when you will study in medicinal chemistry. That is the simple structure of histamine. From that picture you can see that is consists of a ring, that is NH5 ring. That ring is called as imidazole. So histamine is consists of a ring that is imidazole. So that histamine is imidazoline ring containing AUTOCOID. So this group is called as imidazole, and that remaining, that is one carbon, that is second carbon, and amino groups are there. So that is called as 1,2 means ethylamine. So this is the simple introduction about histamine. What is that? Histamine is a AUTOCOID which plays an important role in pursuing different physiological action that I am going to discuss into the upcoming slide. That is a amino derived AUTOCOID which is generally belongs to or derived from a amino acid called histidine. That's why that is amino derived amino AUTOCOID. So that is also called amino derived AUTOCOID because it belongs to the amino acid histidine. That is the simple structure of histamine, which is consist of imidazoline ring and two methyl group with amino group. So that histamine is simply consist of imidazoline ring and ethylamine single chain. So that is the simple introduction about histamine. Now I will discuss in that slide about the different site from where generally histamine secreted from or generated from. In the previous class only I have single line I have explained about the different site and physiological actions of histamine, serotonin, prostaglandin, but that was only a single line. From today we are going to discuss in individually as I promised. Today's topic is histamine, so we will study specifically about the site of histamine, then the classification of histamine, physiological action, etc. So now we are going to discuss in the slide about the site of histamine. So from where histamine generally generated or secreted from, that can be from mast cell, and that also can be secreted or generated from the some non-mast cell also. So now we will discuss about which are the mast cells that generally generate histamine. That involves skin, that also involves GIT, and also pulmonary tissues. So these are the mast cells, that is skin, GIT and pulmonary tissue, where generally histamine generate or secreted from, and some non-mast cell from where histamine generally secreted from, that is CNS, then it also can be generated from the blood cell, that is basophil, and it also can be secreted from the growth activating tissues. So from where that histamine generally can be generated or secreted from, that can be secreted from mast cell, that includes skin, gastrointestinal tract and pulmonary tissues, and from non-mast cell that involves in central nervous system, basophils and growth promoting tissues, that mast cells and non-mast cells generally generate and secreted histamine. Now I am going to discuss about the mechanism of release. In that slide, what I am going to explain? I am going to explain how that histamine is going to generate from that mast cell or non-mast cell that I have already discussed into the previous slide. That mechanism we are going to study now. For that, I will draw a simple diagram. That is a cell line. Okay. Here simple one cell is there, that is not that important. Here we are assuming that is a mast cell. Now in that mast cell, intragranular acidic protein is present here. That acidic protein is also constituted with heparin, and in that cell inside different blood cells are there. Generally here it involves neutrophils. Just an example for I am telling, that is blood. Now what it will happen here, protein heparin and acidic protein that is consist of with mast cell will be here, and the histamine will be derived from histidine and will be binded into this mast cell. Now what it will happen, due to the exocytosis of mast cell, that mast cell is going to exude. So during that explosion of mast cell, it is going to release the binded histamine. That is binded histamine. That is binded histamine. So when they are releasing that histamine, that mast cell is going to replace by different immunoglobulins that involve IgE and IgM, and also it will be replaced with Ca2 plus in form of histamine. So what is the release mechanism of histamine? You have to remember that mast cell is consists of a acidic protein and heparin, which is generally a intracellular protein which consists or bind with histidine amino acid. From that histidine amino acid, histamine generally generates. When the exocytosis takes place in mast cell, that mast cell is going to exclude, and due to that explosion, that binded histamine comes out from the mast cell, and for that releasing of histamine, that will be replaced with calcium ion and with some immunoglobulins that involves IgE and IgM. So when our mast cell is going to exclude, that histamine is going to be released out, and when that histamine is going to release out, it will be replaced by calcium ion, Ig immunoglobulin and IgM immunoglobulin. So that was the mechanism of how that histamine is going to release from the mast cell and from the non-mast cell that I have already discussed in previous slide. So now I am going to discuss about the mechanism of action of histamine. In previous slide I have discussed about how that histamine is going to release from their site, that was mast cell and non-mast cell. Now I am going to explain about mechanism of action. That means how that histamine is going to show their action in our body. Then I am going to explain about the different physiological action that generally showed by histamine. So before discussing about all that function, we have to just remember, or we have to just study in the slide about mechanism of action of histamine. For that you have to remember all that chemicals or hormones or any other autocoids that are generally generated in our body to show its specific action, it generally bind with a protein molecule that is generally receptor to show it action. So any chemical, it include hormone, enzyme, everything that show their action generally by binding with receptor. So here also will be the same case. Histamine also will show its action by binding with different receptor. In especially histamine generally bind with histamine receptors. There are different types of histamine receptors that are present in our body. Those are H1 receptor, H2 receptor and H3 receptors. That all are the receptors by which histamine generally binding with and show their various action depending upon on which receptor it is going to bind with, that is H1 or H2 or H3. What they generally shows their action? H1 generally showed their action by increasing the calcium influx or the calcium amount into the mast cell. Then how that histamine will going to show their action by H2? By increasing CAMP production or cyclic adenosine monophosphate production. And now H3, what it will do? It will do the opposite work of histamine and will inhibit the action of histamine by decreasing the amount of cyclic CAMP. Now we are going to discuss what they generally showed the action when they are binding with that receptor. When they are binding with the receptor of H1, they generally causes smooth muscle construction and they generally promote vasodilation. So when there are histamine binding with H1 receptor, it is going to increases the amount of calcium influx that will leads to the increasing in smooth muscle construction and it will promote vasodilation. Now when the histamine is going to binded with H2 receptor, it is going to increases the amount of CAMP production that will lead to the increase of gastric acid secretion. That will lead to the increasing in gastric acid secretion and it will also promote vasodilation. So when histamine is going to bind with H2 receptor, it is going to increase the gastric acid secretion and it will promote the vasodilation by improving or increasing the generation of CAMP or cyclic monophosphate. In case of H3, when histamine is going to bind with H3 receptor, it is going to decreases the release of gastric acid. So H3 is the opposite type of receptor. When it is going to bind with histamine, it is going to do the opposite work, that is decreasing the release of gastric acid by decreasing CMP. So when it is binding with H1, it will increases smooth muscle contraction and vasodilation by increasing the Ca2 plus influx. Then when it is binding to H2, it is going to increase the gastric acid secretion and promote vasodilation by improving the generation of CAMP. And in H3, it will do the opposite work. When the histamine is going to bind with H3 receptor, it is going to release or inhibit the release of gastric acid by decreasing the generation of cyclic AMP. So this slide I have explained about the mechanism of action by which generally histamine shows their different physiological action in our body. After studying of that mechanism of action of histamine by binding with different receptor of H1, H2, H3, now we are going to discuss by binding with all the receptor, which are the physiological function that generally going to be done or executed by the histamine, that we are going to discuss now in that slide. So according to the different part or different side of our body, we are going to discuss about the function of histamine. First, we will discuss what is the role of histamine in case of central nervous system. In central nervous system, generally that histamine is going to increase the sensory binding and that will increase the different emotion sensations like pain, depression, happiness, etc. So what are their function in case of cardiovascular system? In cardiovascular system, histamine is generally going to decrease the systolic and diastolic blood pressure. So in short, histamine in cardiovascular system is generally going to decrease the blood pressure of our body. Third point, in bronchial smooth muscle. In bronchial smooth muscle only I have explained you, when it is going to bind with H1, it is going to do vasodilation. When it is going to bind with H2, it is also doing vasodilation. In case of H3, it is going to do vasoconstriction. So this is the action on broncho smooth muscle. Then we will discuss about the action in gastrointestinal tract. In gastrointestinal tract, generally histamine increases the constriction of GIT that improves the release of gastric acid from the gastrointestinal tract. So that is the function in related to gastrointestinal tract. And in case of uterus, that histamine is going to help during the abortion of pregnant women. So these are the main general action that is going to showed by histamine, and the most important is controlling of allergic reaction. So what are the general function of histamine that I have discussed now? That is, in CNS, it is going to increase the sensory binding and promoting the emotional sensation like pain, depression, happiness, etc. The most important function is it is going to relate or fight against the allergen and going to create hypersensitivity reaction to indicate the body that something wrong is going to happen in our body. That is done by also histamine. Third, in CVS, histamine generally decreases the systolic or diastolic blood pressure. In short, in CVS, histamine generally decreases blood pressure. In case of broncho smooth muscle, when the histamine is going to bind with H1 and H2 receptor, it is going to show vasodilation. Then in case of gastrointestinal tract, when that histamine is going to bind with H1 receptor and H2 receptor, it is going to increase the constriction of GIT tract that will leads to the increasing in release of gastric acid. But when the opposite side is going to happen, that means when that histamine is going to bind with H3 receptor, that histamine is going to decrease the release of gastric acid. So these are the common physiological action that is generally done or generally helped by histamine in our body. So it is also a very important autocrat that involves in pursuing or performing different physiological actions which are very important for our body. Now, whatever I have discussed till the slide, that I have discussed about the action related to the histamine in positive way. That means what are going to done by in our body histamine that we have discussed till now. But into the upcoming or today's now slide, I am going to discuss about the antagonist of histamine. So now we are going to study about the different drugs that is generally taken from outside by us to do the opposite action of histamine by inhibiting the action of H1 receptor and H2 receptor and H3 receptor. That all drugs are called as histamine antagonists or histamine receptor blockers. So that drugs are classified in two parts, that is first generation drugs and the second one is second generation drug. How that generation generally comes from? First generation means the effectivity is little bit high and that adverse effects are also present here together. But in case of generation, effectiveness is very high and adverse reaction is very low or very negligible. So according to that, first generation and second generation drugs are there which are going to do the opposite action of what histamine generally deal to our body. That's why all that drugs are generally called histamine antagonists or histamine receptor, because they are going to inhibit the action of H1, H2 and H3 receptor. So what are the example of those? First generation of drugs are promethazine, and the second example is diphenylhydramine. So what are the first generation histamine antagonists? That are promethazine and diphenylhydrazine, that are going to show their action by inhibiting H1, H2, H3 receptor and act as histamine blockers, and they are first generation drugs because they have effectiveness with some adverse drugs reaction also is not negligible. So to improve their effectiveness and to decrease their adverse drug reaction, then it comes about the second generation drug. So what are the examples of second generation drug? That is cetirizine. And example is chlorpheniramine. So in that slide I have discussed about the histamine antagonists which are going to taken as a drug from our body to decrease or inhibit the action of histamine by binding with H1, H2, H3 receptor and going to decrease the action of H1, H2, H3 receptor. That drugs are classified into two parts, first generation drug and second generation drug. First generation drug is high effectiveness with amount of adverse drug reactions are also there. Example is promethazine and diphenylhydramine. Then to increase the effectiveness more and to minimize the adverse drug reaction, second class or second generation of histamine antagonists come into the picture. Example is cetirizine and chlorpheniramine. So these are the simple example of histamine antagonists that are going to show their action by inhibiting the release of histamine, by inhibiting the action of histamine receptor like H1, H2, H3 receptor. Now we are going to discuss about the adverse drug reaction that is related to the high consumption of histamine antagonist drugs. So what are the adverse drug reactions related to this? One, itching. Why itching? When that allergen is going to enter in our body, that histamine is getting activated, and when that histamine getting activated, it will cause allergic reaction, and we all know allergic reaction, it sometimes causes itching. So that also a adverse reaction when we are going to consume that drug in high doses. Second one is hypotension. We know histamine is generally going to decrease the blood pressure by binding with H1 and H2 receptors. So when we are taking the drug into the high amount for the high period of prolonged period of time, it is going to decrease the BP very low. That can sometimes leads to the hypotension. The third is tachycardia. Sometimes due to the bronchodilation, because I have told in bronchial muscle it generally constricts the bronchial smooth muscle, sometimes it can cause tachycardia due to the high consumption of all those drugs for a prolonged period of time. Fourth is bronchospasm. And fourth is acidity, because I have told you that histamine generally promote the secretion of gastric acid. So sometimes due to the overuse of all those drugs for a prolonged period of time can cause high increase of gastric acid release, that ultimately going to create acidity in our body. So what are the adverse drug reaction due to the prolonged use of all the drug? First one is itching, second one is hypotension, third one is tachycardia, fourth one is bronchospasm, and fifth is due to the increasing gastric acid, it is going to create acidity in gastrointestinal tract. So by this slide I have finished up about the histamine, where I have discussed about the simple introduction of histamine, how it is belongs to or derived from, what are its simple structure. Then we have discussed about the different site, mast cell and non-mast cell, from where histamine generally released from. Then we have discussed about the mechanism of release by which that histamine is going to release from mast cell and non-mast cell. Then we have discussed about the mechanism of action of histamine, where we have discussed about how histamine is going to show their action by binding with H1, H2 and H3 receptor. What are the actions they are going to show, by which it is going to show their action, that also we have discussed. Then we have discussed about their simple general physiological action that generally act by them in CNS, in CBS, in allergic reaction, in GIT, in uterus, etc. Then we have studied about the different histamine antagonist drugs that are going to inhibit the action of histamine by binding with H1, H2, H3 receptor. In that we have studied first generation drug and second generation drug. And at last, in last slide, I have discussed about different adverse reactions that involved in the prolonged use of all these drugs, like itching, hypotension, tachycardia, bronchospasm, high gastric acid release leads to acidity, all this in that video. So I have explained about the autocoid of histamine that is going to play an important role in our body. So in the next class I am going to discuss about all that same details about prostaglandin into the next video. So in next video the topic is prostaglandin. For today, go through that histamine chapter and ask your question, whatever problem you have, and ask me in comment section. I will try to answer them. For today, histamine. From tomorrow I am going to discuss about prostaglandin, then serotonin. Like that we are going to discuss in upcoming classes one by one. Thank you.